Hello and welcome to another episode of Momento Malum today. The American cranberry is one of the few fruit crop species that are native to North America. The major production of cranberry is the United States and Canada, with other production areas being Chile, New Zealand, and Europe. Cranberries receive considerable attention due to the antioxidant status of the species. Dr. Vorsa and Dr. Zalapa will compile the data regarding the genetics and domestication of the cranberry, and here are the results. The domestication of the cranberry was initiated in the 1820s in Massachusetts. The first reported cultivar was propagated and was named Early Black. It was bred by Nathaniel Robbins, where the first vines were selected from a swamp in Harwich, Massachusetts. This occurred in 1852. This cultivar was used in the first commercial planting, which was established in 1857 in Massachusetts. During the 1880s and early 1900s, over 132 varieties were named and propagated from wild selections. These were obtained from Massachusetts, New Jersey, Wisconsin, and Michigan. It appears that the criteria for selection were season of harvest, fruit color, berry size, and productivity. Of the cultivars selected from native populations, the major cultivars utilized were Early Black, Howe's, McFarland, and Searles. These were the major cultivars in the early to mid-20th century. Early Black, Howe's, and McFarland were selections from Massachusetts, and Searles was a selection from Wisconsin. The first breeding and selection cycle was initiated in 1929 by the USDA in response to a devastating disease called false blossom. This disease is caused by a Phytoplasma bacterial species. The first selections were planted in New Jersey in 1934, and new crosses were added in 1937 and 1943. These were bred from 30 crosses made from 18 native selections, which led to the creation of 8,692 seedlings that were evaluated at White's Bog Research Station. The majority of the parents were selections from wild populations from Massachusetts, but there's a few cultivars utilized in the crossing process. These include Prolific, Searles, McFarland, and Potter. Of the 30 crosses, McFarland was a parent in 16, probably because of the larger fruit size of the specific cultivar. The first generation of selectively bred cultivars were created in the 50s and 60s, and later selections will be bred in the 70s until the late 2010s. Most of these cultivars were bred by various public universities and the USDA, but a few were bred by private companies like the Valley Corporation. If you want to donate to my channel, the links are below. If you want to buy my book, you can also find that link in the description box below. On a genetic level, the populations of wild cranberry in Wisconsin are the most genetically diverse populations of wild cranberry. Overall, however, the wild populations had a low genetic diversity despite the regions being mostly untouched. This makes sense, however, since the cranberry reproduces both through sexual reproduction and by cloning itself. As a result, the populations of wild cranberry are a mix of clones and seed-produced progeny, with some populations consisting mostly of clones, and others consisting mostly of seed-produced populations. False blossom is the main disease in cranberry cultivation, and the cultivars Henry, Berlin, Metallic Bell, Palmater, Prolific, Bennett, Pride, Early Black, and McFarland have different degrees of resistance to this disease. Another major disease is fungal fruit rot, and several lines have been found to be resistant. These are Blackvale, Howe's, Matthews, Shaw Success, Stevens, Wilcox, Bugel, Stankovich, Bud's Blues, Holston, Cumberland, and US 89-3. In terms of root rot, there's very few cultivars that have resistance, and only 3 were found to have resistance. These are Stevens, Franklin, and Bergman. And that about covers everything. Thank you for watching.